Morning.
Good morning, Doc.
So it's already 8 o'clock, so we will start. Okay. So the topic today is about your ortho, okay, your orthomyxoviruses. This is otherwise known as your influenza, okay, influenza viruses. Highlight. Okay, let's go to important properties of your okay, orthomyxoviruses. Now, your orthomyxoviruses comes in usually spherical, okay, but some of them assumes several shape, okay, otherwise known as your pleomorphic. But majority of your orthomyxoviruses is in spherical in shape and is around 80 to 120 nanometer in diameter. So this is bigger as compared to your picorna viruses, because your picorna is usually the smallest among all types of viruses, and the largest among all type of viruses usually your rhabdoviruses, which is just like a bullet shape. Now, the composition of your orthomyxoviruses, usually the protein is 73%. Okay, 73%. And these protein are usually, there are 9 structural protein and 1 non-structural protein. Okay, so majority of the content of the orthomyxoviruses are composed 73%. Okay, are your proteins, then followed by your lipid, okay, which is 20%. And this lipid is usually the one coating, okay, or the one coating the envelope, okay, envelope, content of the envelope. And your carbohydrate is around 6%. And one of the carbohydrate is usually the sialic, okay, or the glycan portion of the virus. And we have a portion of which is your RNA, so 1%. Okay, so your orthomyxovirus is a single-stranded negative sense RNA. Okay, and this is usually segmented. Okay, segmented. So this is the single-stranded negative segmented negative sense RNA, and it has two important structures in the envelope. Okay, and these are your viral hemagglutinin and your enzyme neuraminidase. Later on, we will discuss what is these two. Okay, antigenic structure of your orthomyxovirus. Antigenic because this is the one where your antibody binds and producing, okay, antigen antibody reaction. That's why they are antigenic, okay, particularly your hemagglutinin, okay? And the replication, okay, usually happens in the nuclear transcription. Side said usually the RNA usually replicates in the cytoplasm, but in this case, okay, usually it replicates in the nucleus. Okay, so we have later on the Cap5 prime terminal of the cellular RNA scavenge as primers, and particles mature, okay, by budding from the plasma membrane. So once they mature, ready to be released, okay, but before they are released, they are usually bound to the plasma membrane, okay, to the plasma membrane, and usually the neuraminidase usually cut, okay, this virus in order for the virus to be released to infect another cell. Okay, so they by bud. Okay, so they are removed by the neuraminidase for the virus, for the new virus to be released to infect another cell. Okay, so one important properties of your orthomyxo is your genetic reassortment. So it can change its structure, it can change the structure of your hemagglutinin, okay, so that it will not be recognized by the antibodies. That's why there is what we call reassortment. Same true with the SARS-CoV-2, okay, the spike usually changes its structure so that the antibody or the vaccines can no longer, okay, can no longer recognize this. That's why we need a booster dose of this vaccine, okay? And eventually, okay, this COVID-19 vaccine is still underway and study if we need booster dose, and if we need booster doses, how long, every 6 months ba or every year. So we will wait for the study. And the influenza virus causes worldwide epidemic. Okay, worldwide epidemic meaning it can cause pandemic. Okay, just like what we have the Spanish flu. Okay, the Spanish flu way back in 1918 where millions of people died. Okay. Now, your influenza, you have three serotypes, your influenza A, your influenza B, and your influenza C. Okay, your A and B occur eight separate segments. Okay, eight separate segments, while your C virus contain only seven segments of RNA. Okay, so your C does not have the enzyme neuraminidase gene. Okay, then the sizes and protein coding assignments are known for all the segments. Okay, and most of the segment code for a single protein. Okay. Now, the influenza virus particles contain nine different structures, okay, structural protein. Okay, so we have nine structural protein and one non-structural protein. So the NP, otherwise known as the nucleoprotein, okay, associates, okay, in connection with the viral RNA to form what we call your ribonucleoprotein, okay, structure, which is around, maliit lang siya, 9 nanometer in diameter. So this nucleoprotein, okay, are also, okay, assumes a helical configuration. So para siyang pillow, spherical, okay, configurations. Later on, we will see the structures of these and form what we call your viral nucleocapsid. Okay, viral nucleocapsid. Okay, now there are three large protein, very, very important for your transcription and replication of the virus. And these are your PBB1, PBB2, okay, and your PA. So these are three large protein responsible for the transcription and replication of this virus to form a new virus. So they're very, very important for the production, okay, of your virus. So we have your matrix 1 protein, okay? So this matrix 1, a very important structure, okay, forms the shell underneath the viral lipid envelope. Okay, so it coats also the viral envelope. Okay, so this is important in the morphogenesis and major component of the variant. Okay, so majority, okay, 40%, okay, of your viral protein is made up of your M1 protein. So later on, we will see the structure of this M1 protein. Okay, next. Two important structure in the envelopes are what we call the heme agglutinin, okay, the HA, agglutinate because it agglutinates with the erythrocytes, okay, the red blood cell. And we have the enzyme neuraminidase, the NA, okay, so they are inserted into the envelope, okay, and they are exposed as spikes. So the spikes are termed as your hemagglutinin, okay, and your neuraminidase. In the SARS-CoV-2, we simply term them as spikes. Okay, so these are important antigen, okay, that determine antigenic variation. So it assumes or it has different. It can do what we call the genetic reassortment. So nag-iiba yung genetic composition niya. So we call it antigenic variation. So it varies. Okay, influenza viruses and host immunity. Now, which is more abundant, hemagglutinin or neuraminidase? So your hemagglutinin is around 25%, while your enzyme neuraminidase is about 5%. So mas marami ang hemagglutinin. So because of the segmented nature of the genome, okay, when the cell is co-infected by two different viruses of a given type, okay, so two viruses of a given type, for example, virus coming from the animal, okay, and influenza virus coming from the animal, and a virus from a human, when they bit together, it undergoes what we call genetic, okay, reassortment. So there will be mixture of parental gene from the animal, okay, and from the human. Okay, so maybe assembled into progeny virion. So they make a virus, a new strain of virus. And this phenomenon is what we call genetic reassortment. So genes, influenza genes from this animal goes into human, okay, and there will be genetic reassortment. Okay, and they may result in sudden changes in viral structure antigen. So it may change the shape. For example, it's usually spherical. Okay, the spike is spherical and it may change into semi, okay, semicircle. Okay, so this semicircle can no longer be recognized by your antibody. Okay, it is usually recognized by the spherical hemagglutinin, but when it changes the shape, it can no longer be recognized by the antibody. So there will be genetic reassortment and may result in viral surface antigen structure change. So this is the property that explains the epidemiologic features of influenza, okay, and poses a significant problems for vaccine development. So it can no longer be recognized by the vaccine, okay, because of the genetic reassortment. So that's why there is annual, okay, dose of your influenza based on the isolate which strain of influenza virus. Now, this is the electron microscope typical feature of your influenza virus. So this is usually spherical, and sometimes it is usually pleomorphic. It may assume several shapes. And you see the outer structure, and these are your spikes, okay, made of your hemagglutinin, okay, hemagglutinin and the neuraminidase, and inside here are your structural and non-structural protein. So again, this is the cross-section of the influenza virus. Okay, so this is the envelope. Okay, within the envelope, there are your glycoproteins known as your hemagglutinin and your neuraminidase. And this is the nucleocapsid. And within or inside the nucleocapsid are the viral, okay, are the structural protein, yeah, the RNA as well. Okay. Now, in terms of classification and nomenclature, okay, your influenza A, yeah, A contains human and the animal strain. So it can be pigs, swine, ducks, or avian, okay, animals. Okay, it may reassort, okay, doon sa humans. And this is usually common in influenza type A. Okay, your influenza virus type B, okay, contain human strains of type B, while your C may contain influenza type C, okay, virus of human, and also swine. Okay, also swine. Okay, so kaya nagkaroon din dahil, di ba, meron kang swine, okay, African swine. Okay, now this is again the structure of your influenza. Okay, see, this is the section of your influenza. This is the hemagglutinin. The yellow one is the hemagglutinin. It has 3 heads. Okay, so it's usually trimeric. This one, two, three. Okay, and in this head contains the active site. Okay, the active site where the antibody usually binds. For example, here there is an antibody. It binds here into the active site of your hemagglutinin. Okay, and this one are your neuraminidase. Neuraminidase. So they are attached, okay, into the plasma. Yeah, yeah, some plasma sa envelope. Yeah, this is the envelope. They are attached. Okay, you can see here the red one, okay, known as the M2. M2, okay, is responsible for the ion channel protein. Okay, ion channel protein where your ion usually enters. Okay, and you see the green one, okay, the green one, okay, are your M1, okay, M1 protein, okay, 40% is made up. So it's linked here. There's an envelope. Yeah. Okay, so this is the and your neuraminidase usually 4 heads, 1, 2, 3, 4. So your neuraminidase is usually tetrameric. Okay, so antigenic difference exhibited by 2 internal structure of proteins. Okay, 2 internal structure of proteins. We have the nucleoprotein and the matrix M protein. They are used to divide influenza viruses into type A, type B, and type C. Okay, so this protein possesses no cross-reactivity among the 3 types. So meaning your influenza type A cannot cross-react with your type B, while your type B cannot cross-react with type C. Okay, or your A cannot cross-react with C. Okay, and vice versa. So antigenic variation in the surface, yeah, this one. antigenic variation in the surface of your glycoprotein, okay, your hemagglutinin and neuraminidase are used to subtype viruses. I think you have heard yung mga H1N1 during the Spanish flu, okay? So, and only A has designated subtype. So only A has designated subtype like H1N1. Wait lang po. Okay, so next one. So this is how we name, okay, your influenza, a strain. So we have what we call H1N1 type A flu virus of your duck origin. So galing sa duck, okay, isolated in Canada, Alberta, and your H1N1 was the 35th strain discovered, okay, in 1976. Okay, so how do we term this? Okay, your influenza type is type A, where the host of origin is duck. Okay, where did I isolate? It's in Alberta. This is the 35 strain, okay, H1N1. Okay, when did they isolate? For example, 1976. And the subtype, okay, of influenza A is known as your H1N1. Okay, so this led to your Spanish flu pandemic. So your antigenic descriptions of your hemagglutinin and Na are given in parentheses for type A. Okay, this one. So there should be here a parenthesis. Yeah. So the host of origin is not indicated for human isolate. Okay, some of which are like type A, where did they isolate? Hong Kong. Okay, so what strain in the three strain? So isolated 1968, and this the parenthesis H3N2. Okay, so but others, okay, indicated for others such as meron dito nakalagay na swine. Okay, so type A, galing sa swine. Okay, where did I isolate? Iowa. Okay, so this is the 15th strain. Okay, among the 30 strain. Okay, otherwise they isolated the subtype H1N1. So there is a parenthesis there. Okay, so far, how many subtypes of HA? Usually, okay, there are 15 subtypes. So H1, H2, H3, H4 to H15, and 9 subtype of N1 and N1 and 2 to N9. So H1 can combine with N1, so that's H1N1, or H3 can combine with your N2, otherwise known as your H2 N2 or H3N2, or your H5 can combine with N1. So you have your H5 and 1. Okay, so depende where would this be isolate, kung saan makukuha ang countries, and when did this happen. So in many different combinations, so many different, so H1 combined with N1. So been recovered from birds, animals, or human. Okay, so yung mga genetic reassortment. So for 4HA, okay, H1, H2, H3, and H5, and 2NA and 1 and N2 subtypes have been recovered from humans. Okay, now as I've said, this is your structure of your hemagglutinin. So it's a trimeric, so 1, 2, 3, and these are the active site where the antibody binds. Now it's important the antibody binds here in the active site para it can no longer bind to the respiratory receptors, respiratory epithelial cells. Just like your SARS-CoV-2, it has to bind to the spike. The antibody has to bind to the spike so that the spike can no longer attach to the ACE2 receptor. Okay, so this is the neuraminidase. It's tetrameric 1, 2, 3, 4. These are the active site where the antibody binds. Okay, so again, these are the structure. Okay, so this is your hemagglutinin. Okay, this one, and this is your hemagglutinin. These are your neuraminidase. Okay, so you see this one, the structure here. Okay, so these are your PA, PBB1 and PBB2, necessary for the transcription and translation of your viral RNA. So these are your viral RNA. Okay, so these are the nuclear export protein, this one, nuclear export protein, or the ion channel. Okay, and you have, sorry, the membrane protein too. These are the ion channel. Okay, so this is your structure inside the influenza virus. Okay, now the structure and function of heme agglutinin. You see here, this is the plasma membrane. Okay, this is the receptor. So the receptor is made up of your carbohydrate glycan. You see here the heme agglutinin. The heme agglutinin binds to the glycan receptor of the plasma membrane so that this virus can get internalized and get endocytosed and pupunta inside the cytoplasm. So the hemagglutinin protein of influenza binds to particle susceptible cells, which are the glycan here, okay? And your hemagglutinin is a major antigen against neutralizing protective antibodies. So see, okay, this antibody will neutralize this, okay, antigen so that it cannot bind to our cell. Okay, so we have variability in hemagglutinin is primarily, so I said there is genetic assortment. There will be variation in size, in shape, okay, or the phenotypic, okay, phenotypic appearance of the hemagglutinin may change. Okay, and this is responsible for the continual evolution of new strains, okay, and subsequent influenza epidemic. So your hemagglutinin derived its name from its ability to agglutinate erythrocytes under certain conditions. Okay, so this is a good picture. Okay, you see here your PBB1, PBB2, okay, it's one. Okay, so they are one responsible for the transcription and translation of your structural proteins. So these are the genomic viral, okay, genomic viral RNA. This is the M2. Okay, this is the neuraminidase kasi tetrameric. Okay, trimeric, the heme agglutinin. This is the matrix M1 protein. This is the M2. So this is a good picture of your influenza virus. Okay, now this is your heme agglutinin. Okay, so get a portion of this. This is the heme agglutinin. Okay, so this is clipped into two subunits. So we have subunit A1 and you have the subunit A2. Now your A1 and A2 are attached via your disulfide, okay, via your disulfide bridge. Okay, so this is the bridge, okay, that attach your A subunit 1 and A subunit 2. Okay, now at the end of the subunit 2, you have here the hydrophobic membrane domain. Okay, so the hydrophobic membrane domain, and you have the hydrophilic cytoplasmic domain, okay, near the carboxy terminal, okay, near the carboxy terminal. So your hydrophobic stretch, so your hydrophobic stretch near, okay, it stretch near the carboxy terminal, okay, of your H2, okay. So, and we have also the short hydrophilic tail. So this one, the short hydrophilic tail extending into the cytoplasm. Okay, so they may extend, if they bind, they may extend into the cytoplasm, this one. Okay, so if they bind, they may extend into the, okay, they may extend into the cytoplasm. Okay, so this is your neuraminidase. As I've said, it's usually trimeric, and the A1 are the active sites where the antibody binds. Okay, so this is the protease cleavage. Protease cleavage. Okay, so dito magki-cleave. Okay, so if there is a protease cleavage, okay, it makes the virus more infectious. Okay, it makes the virus more infectious. So this is the structure of your hemagglutinin. The virus provides, okay, or the surface of infected cell. Okay, so this is the plasma membrane. Okay, so it binds to the sialic acid. okay, sialic acid, and it may extend into the cytoplasm. Yan, extend into the cytoplasm. Kanina pinakita natin. Okay, so your letter D here, okay, letter D, this is the structure of your neuraminidase, this one. Okay, so the structure of your neuraminidase, it has a stalk, okay, and at the top of your are the box, the tetrameric boxes with the active site. So extend into the cytoplasm. This may extend into the cytoplasm. Okay, so this is again the functions of your M1. So your M1 is on M1. Okay, interacts with the ribonucleoprotein. So this M1 may interacts with the ribonucleoprotein. So we have the M2, okay, responsible for the ion channel, the sodium, potassium. Okay, you have the PBB1, this one, PBB1. Okay, it has the enzyme endonuclease. Okay, responsible for the elongation of your RNA polymerase subunit. You have the PB2. This is the cup region. Okay, the PBB2, this is the cup region from the 5' to the 3'. Okay, and the red ones, okay, see the red ones, the RNA polymerase, this one, the RNA polymerase. Okay, and the nucleoprotein. Okay, so as I've said, there is a cleavage between the H1 and the H2. And this cleavage is necessary for virus to be infectious. And it's mediated by the cellular protease one. Okay, so this one is the cleavage. Okay, so proteolytic activation of the enzyme protease, this one, proteolytic cleavage. So once there is cleavage, it makes the virus more infectious. And this is mediated by your enzyme proteases. Okay, and this is basically located, okay, in the respiratory tract. Okay, so in the respiratory tract, you have protease enzymes that cleave the HA, so making them more infectious. Okay, so they will infect the epithelial cells of the alveoli, okay, destroying them. Okay, and commonly at only at two sites, dito sa respiratory tract, yung undergoing a proteolytic cleavage for virus to become more infectious. Okay, so the antigenicity of your neuraminidase and other glycoprotein on the surface of influenza virus particle is also important in determining the subtype of influenza virus. Diba? Your N1, N2, N3, okay? They also determine subtypes. Okay? So this is the hemagglutinin. Okay? So this is a tetramer composed of 4 identical monomer. One monomer, one. 4 identical monomers. So there is a slender stalk here. Okay, on top of each is the box shape. So there is also a catalytic site for your Na on top of each head, so that each Na spike contains 4 active sites. So this is your neuraminidase. So there is an enzyme here. So your neuraminidase function at the end. Sabi ko nga, at the end, for example, this is the cell, and this is a new virus. Okay, the new virus ready to be released. Okay, so the enzyme will cut the new virus from attaching to the plasma membrane for it to be released. So there is a sialidase enzyme, okay, that remove the sialic acid from the glycoconjugate. So parang hindi sila mag-aggregate, the sialidase enzyme, okay, is released to release is released in order for the virus to exit from the plasma membrane. So it facilitates the release of the virus from the infected cell. Later on, we will see how they are released, okay, during the budding process, and helps prevent them self-aggregate. Para hindi sila mag-aggregate dun sa plasma membrane, it has to be released, okay, through the enzyme sialidase by the neuraminidase. It is possible that the neuraminidase help the virus negotiate through a mucin layer in the respiratory tract, reach the target epithelial cells. So let's go to 2 important nomenclature in influenza virus. So we have what we call the antigenic drift and the antigenic shift. Now, in terms of drift, okay, these are usually, okay, minor changes, okay? Minor changes in the drift that undergo what we call your point mutation. So nag-iiba yung gene, Okay, so nagkakaroon ng point mutation. So there is usually minor antigenic changes. Okay, and when it is usually a major antigenic change, ibig sabihin major, it changes the shape of the heme agglutinin such that can no longer be recognized by the antibody. Instead of a globular, becomes semicircle. Okay, so that result in appearance of a new subtype, and this is usually causing an epidemic or worldwide epidemic, otherwise known as your pandemic. So there is usually major change in heme agglutinin or neuraminidase. Minor is usually your antigenic drift. Okay, so the mechanism for shift is what we have is genetic reassortment between human and influenza virus. So ang human may HA and NA. Ang swine, for example, ang avian, okay, birds meron din HA and N1. So they may change, okay, together, okay, they may reassort or combine, okay, leading to antigenic shift. So your B and C do not exhibit antigenic shift, only A that exhibit antigenic shifting because few related virus exist in animals. And besides, okay, so sa C, hindi gano'n nagtatagal kasi namamatay naman din kaagad yung mga ipon. Okay, so this is the replication. How do they replicate? So this is a schematic diagram, okay, of the life cycle. See the life cycle of the influenza virus. Okay, so after a receptor, this one receptor mediated endocytosis. Okay, and there will be the release of your RNA, okay, the nucleocapsid, and this RNA goes into the nucleus. Okay, with the aid of these PBB1, nucleoprotein, and PB2 undergo transcription and translation in the nucleus. Okay, and there will be formation of your hemagglutinin and neuraminidase, okay, particularly in the endoplasmic reticulum. Okay, so your viral attachment, this one, the virus attaches to the sialic, okay, portion of the plasma membrane. Okay, so via the receptor mediated, mediated, located on top of a large globule. Di ba kanina may active site, it binds here to the sialic, large globule of your hemagglutinin. Okay, and it's undergo endocytosis, coated vesicle. And this vesicle interact with the lysosomal, okay, and the lysosome releases its enzyme, okay, which has an acidic pH. So once it releases the enzyme, the ribonucleic acid, okay, nucleocapsid are released. Okay, once they are released, they get inside here the nucleus. With the help of your PBB1, it undergo transcription. Okay, transcription. So later on, you'll see this what's happening in step 1, step 2, step 3, and step 4. So the mRNA, this one, are produced from the viral nucleocapsid. So the virus encoded, okay, we have the enzyme polymerase consisting of complex of 3 protein. The NP, the PBB1, the PBB2 is primarily responsible for the transcription. This one, the N1. Okay, they will be responsible for the transcription of this viral RNA to produce, okay, a lot of your RNA. Okay, so usually the virus matures. Okay, so when there is one here, the virus, kapag okay na, there is production of the new virus. So for example, this is the plasma membrane. Okay, and this is a new virus. This new virus will attach here to the plasma membrane. Yeah, yeah. New virus to the plasma membrane by budding. So they by bud from the surface of the cell. The enzyme sialic acid is released. They will cut this one, and this new virus will be released to infect another cell. Okay, so the nucleocapsid are assembled in the nucleus. So this one, they will be assembled. The nucleocapsid will be assembled here in the nucleus. Okay, and move out of the cell surface. So the hemagglutinin and the neuraminidase are synthesized. This one, they are synthesized in the endoplasmic reticulum. This one, so they are synthesized in the endoplasmic reticulum with the help of the ribosome. This one, This is the ribosome. So they are transcribed from 5 prime to 3 prime to make a protein, hemagglutinin and neuraminidase. So it happened in the endoplasmic reticulum. Okay, so the M1 is one, the M1 is one, yeah, M1, so the M1, okay, serve as a bridge linking the nucleocapsid, this one, linking the nucleocapsid here, okay, ends of the glycoprotein. This is the M1. It's linked to the nucleocapsid. So the Na, this one, the Na removes, okay, it removes the terminal sialic acid from cellular and viral surface glycoprotein, facilitating the release, yeah, release of the virus particles from the cell and preventing their aggregation. So para hindi sila mag-aggregate, okay, the neuraminidase releases the enzyme so that this will now be released. Okay, so this is the step 2. Okay, so this one, the step 2 is that step 1 is the attachment. Okay, step 2, the messenger RNA. This one, the messenger RNA are exposed, okay, to the cytoplasm. This one. So this is the messenger RNA, yeah, exposed into the cytoplasm. Yeah, this one. This is the cytoplasm. This is the nucleus. Okay, you have here the step 3. Okay, so early viral proteins required for replication and transcription, including the NP, this one, the NP, okay, and the PBB1, okay, are transported back into the nucleus, okay. So the RNA polymerase activity of the PBB1 protein synthesizes positive, yeah, it synthesizes a positive single-stranded RNA from a genomic negative single-stranded RNA molecule. So para makapag-produce sila ng maraming RNA. Okay? So in step 4, the positive single-stranded RNA template are copied by the RNA polymerase activity of the PBB1. Okay? And the step 5 here, some of these new segment serve as a template for the synthesis of more, this one, synthesis of more viral mRNA. Later in the infection, they become progeny genomes. Okay, so parang immature na virus, progeny genomes. So the viral RNA molecules are transcribed from some genomic segments, encode the structural protein such as this one. It will encode the structural protein, okay, and this protein will be synthesized, okay, into the endoplasmic reticulum with the help of the ribosome. Okay, so after which they make a new progeny of virus, and this virus will attach here. Okay, so magpo-form na yung virus, mag-assemble na dito, parang Voltes 5. Okay, yung virus magpo-form parang Voltes 5, and they are attached into the plasma membrane. This one. Yeah, this one. Attached into the plasma membrane. Okay, and the viral genome segment are packaged as progeny virions from the cell, okay, and they did now to be released to infect another cell. Okay, so in 6, packaged as progeny virion bud from the host cell, and the enzyme neuraminidase will be released in order to prevent this from aggregating here. Okay, so this is a good table in the Jowets where we differentiate, okay, certain diseases. Now, your influenza A virus, it has many subtypes. So H1N1, H5N1, H3N2. Okay, because of these different serotypes, okay, there is no immunity, lifelong immunity. So that's why, okay, so we need a booster yearly. So there is no viral latency. So we have here your para-influenza causing group. There are many subtypes. So usually if there are several subtypes, there is no immunity to it. Okay? And there is no latency. Unlike your herpes, there is usually latency. So your rubella, so there is a vaccine here. So that's why there is lifelong immunity. If you have rubella infection before, you can no longer have German measles. Okay, you see the rhinovirus, as I've said, there are around 100 or 150 serotypes. So that's why there is no lifelong immunity. In mumps, there is vaccine, MMR. So that's why there is lifelong immunity. So Coxsackie, wala rin. Coronavirus, there is no lifelong immunity. Sa common colds. You see the herpes virus, diba? It remains latent in the dorsal ganglion. That's why there is viral latency, but there is no lifelong immunity. But in terms of varicella, chickenpox, there is lifelong immunity. Epstein-Barr, there is lifelong immunity. So all of this has viral latency, okay, including the adenovirus. Okay, let's move on to your influenza infections in humans. So this influenza is usually transmitted through a droplets or even airborne. So when a person sneezes, infected person sneezes, okay, going into this susceptible individual to may acquire through okay, droplets or airborne or through inanimate objects holding contaminated. When a person cough out, okay, and this okay virus gets into the commonly attached okay doorknob, and this one usually touches his nose or eyes, it may enter. So once it enters okay, into the respiratory, okay, this one, the virus enter into the respiratory tract. Okay, it binds here in the sialic acid. Okay, it is now endocytose. Okay, so once it endocytose, it attaches here to the epithelial cells of the alveoli. Okay, once it attaches, since this is foreign, okay, it undergo or release a lot of cytokines or your inflammatory response. There will be tumor necrosis factor, interleukin 1, interleukin 2. So this will call your anti-inflammatory cells like the macrophages, okay? So macrophages to phagocytose this influenza. But then, okay, it undergoes epithelial damage and inflammation, later on inflammation, okay? So this is how severe also your influenza, and later on, this may cause secondary bacterial infection. Okay. So this is the incubation period. So incubation period usually is 1 to 4 days. But prior to the symptom, you are now shedding the virus. So a day prior to the symptoms, you may be shedding the virus, okay, to your household member. And it usually peaks within 24 hours and remains elevated for 1 to 2 days. And you may no longer be infectious after 5 days. On the sixth day, you may now be considered as a decline over the next 5 days. So these are the symptoms also. You have sore throat, dry cough, rhinitis, fever, chills, headache, okay, fever. These are the flu signs and symptoms. This one, the virus gets into you. Okay, so one important innate immune response is known as your interferons. Okay, so they are detectable immediately after viral infection. So this is part of the innate immunity response which contribute to host recovery from infection. Remember, interferon, this infected cell releases interferons in order for the nearby cell, the virus no longer replicate. Okay, so there will be specific antibody and cell-mediated response cannot be detected for another 1 to 2 weeks. So ang tagal pa ng cell-mediated response cannot be detected in 1 to 2 weeks. So this is the summary in the pathogenesis of your influenza. It enters the respiratory epithelial cells via the hemagglutinin. Okay, in see how fast they replicate in around 4 to 6 hours. They replicate. Okay, so new viruses are formed. So they and then the alveolar cells or the epithelial cells become necrotic. Later on, they desquamate. Okay, and they alter the respiratory defense mechanism. So they alter the phagocytic function of the macrophages. They will alter the ciliary function, okay, and systemic features due to induction of cytokines. So patient may experience fever because of the release of your interleukin and tumor necrosis factor. And antibodies are formed to hemagglutinin, and they are usually protective. And antibodies to neuraminidase can usually limit the viral spread. Okay, so antibodies, para di ba hindi na-cut ng neuraminidase in order for the virus, in order not to be released to infect another cell. So it will limit the viral spread. So what are your clinical findings? Okay, usually the fever usually lasts 3 to 5 days. So as the systemic symptoms like fever, headache, body malaise. Okay, so respiratory symptom typically last for another 3 okay, to 40 days. So mawawala na yung fever, pero yung respiratory symptom may persist for at least 3 to 4 days. Okay, and the cough and weakness may persist pa. Okay, 2 to 4 weeks after the major symptoms subside. So there's still cough and body weakness, 2 to 4 weeks. So this is what we call your uncomplicated influenza. But what happens if the patient suffered from influenza pneumonia or viral pneumonia? So there will be an increased mucus secretion which help carry agents into the lower respiratory tract. So by gravity, mucus yan, bababa yan to the lower respiratory tract, causing your pneumonia. So there will be dysfunction of the phagocytic cells. So the alveolar macrophages, the white blood cells can no longer function, okay, because of impaired, okay, response because by this influenza. There will be loss of ciliary clearance, okay, and provision of rich. So there will be rich or secondary bacterial infection. And one of the most common causes of this bacterial infection is your Staphylococcus strain, which secrete protease able to cleave. Remember, the protease make them more infectious. So once there is secondary bacterial infection, it may be more, or it can be more infectious. So allowing production of a much higher titers of infectious virus in the lungs. So children who are around or 2 okay, to 16 years of age with influenza symptoms should not be given with aspirin, because children with influenza, when it's given with aspirin, it may lead to what we call Raynaud syndrome, and this is an acute encephalopathy, okay, among children and adolescents. So viral infection should not be given with aspirin because the mortality is very high, 10 to 40%. Okay, so what's immunity? Okay, your immunity to influenza is long-lived and subtype specific. So there is a vaccine which is subtype specific. Okay, so antibodies against HA and NA are important immunity to influenza. Okay, they are protective. So resistance to initiation of infection is related to antibodies against your HA, but decreased severity disease, okay, and decreased ability to transmit virus to contacts are related to antibody directed against the neuraminidase. Okay, so antibodies against the ribonucleoprotein are type specific and are used while in typing viral asymmetry for A and B. So this is the virus and this is the antibody. Okay, so your immunity, serum antibodies persist for many months to years. Okay, so antibody also modifies the course of illness. So just like the vaccines against COVID-19, it modify the course of illness. So it will prevent the mild, okay, or moderate to become severe or critical pneumonia. So it modifies, okay, the course of illness. So a person with a low titer of antibody may be infected, but will experience mild form of disease. Okay, so antibodies like IgA is very, very important in the respiratory tract, but this is not usually lifelong. So they are only in the local secretory, okay, in preventing, okay, viral infection. So of shorter duration. Unlike our IgG, this is more of a longer, okay, immunity. So there is no protection against 3 types of influenza. So a person with pre-existing antibody to original strain, okay, mild infection with new strain. Okay, so we have antigenic sin. So this is what we call subsequent infection or immunization reinforce the antibody response to the first subtype of influenza experienced years earlier. So you develop also memory cells. And when you experience this same strain, okay, you may have only mild infection. Okay, so what is also important is your cell-mediated response. But I've said this is not seen in the next 2 to 3 weeks. So your cell-mediated response is important, particularly your cytotoxic T cells or your killer cells, because these killer cells will remove the viral infected cells. So para silang janitor that will remove the viral infected cells. Okay, so how do we test or diagnose? Okay, test or diagnose your influenza. So the most common test, of course, is still your PCR, polymerase chain reaction. Okay, so this is a PCR that detects, okay, several strains of your influenza. So it may be H1N1, H5N1, or H5N2. Okay, so ano yung gene na na-amplify niya? If it amplifies your H1N1, then the patient is infected with H1N1. If the gene is amplified by H5N1, okay, and the person is infected with H5N1. So this is the very, very sensitive and specific. So you have multiplex rapid detection, okay, multiple pathogen in a single test. For example, in a single test, it can detect SARS-CoV-2, influenza, adenovirus, para-influenza. So there are multiple viruses that can be detected by your PCR. Okay, and it's very, very expensive, around 23,000 in single test. So rapid tests less than 1 day using reverse transcriptase polymerase chain reaction are preferred for diagnosis of influenza. But in SARS-CoV-2, we only detect the SARS-CoV-2, the RT-PCR. Okay? So wala tayong respiratory panel. Only in tertiary centers like St. Luke's, Makati Med, they have this respiratory panel which can detect 16 virus or more. So second is you can do culture, viral culture, but this is very often fastidious and grows slowly. So it's really time consuming and it's too cumbersome to produce this. We do not do here kasi medyo matagal, but we have still here the PCR. So they are held at 4 degree, freezing and thawing reduces the ability to cover virus. Okay, if storage time will be exceed 5 days, the sample should be frozen at -70. So it lasts 3 to 10 days, and trypsin, which cleaves and activates the hemagglutinin, so that replicating virus will spread throughout the culture. So they add your trypsin for the virus to replicate. Okay, serology, okay, your antibodies, okay, to several proteins are produced during infection. So the immune response against HA glycoprotein is associated with resistance to infection. So for your antibodies to become what we call your neutralizing antibodies or protective antibodies, there must be at least fourfold or greater increase in titer. Okay? So dapat mas mataas. Okay? Fourfold. Okay? So ELISA is more sensitive than other assay. So fourfold increase in antibodies for it to become neutralizing antibodies. So this is what we call your fourfold. Okay, so this one, the regeneration and distribution of media here, then the distribution of serum from the patient here, again a serum. Okay, they will incubate this tray, and after which they will read. Okay, so wala siyang change in color, only orange, red, or fuchsia, absence of antibodies. But if this one, wala pa yung antibodies, and if it changes 1:2, pero meron din sa 1:4, there is still change in color in 1:8, and there is change in color in 1:16. So 1, 2, 3, 4. So there is fourfold increase of your antibody. So there is considered as a protective antibody. So represents a high titer of serum antibodies. Okay, so this is protective of your influenza strain. What type of strain? So, yan, fourfold. Yan. 1, 2, 3, 4. Hanggang 4 nag-change yung color, then you are protected against this influenza strain. So the epidemiology is, of course, you have in the northern and southern hemisphere part of the world, okay, which is during the winter, your influenza is usually common. But here in Philippines, which is a tropical country, your influenza is usually present throughout the year, in tropical countries. So again, the reservoir is the humans and animal, okay, common sa type A, okay, so this is usually happening, your antigenic shifting usually happen in type A, so the contact, direct or indirect, and it can be droplet or airborne. So the temporal pattern usually peak in December and March in temperate climate. Okay, incubation period usually 1 to 3 or 1 to 4 days, and they are communicable or infectious a day before to 5 days after the onset in adults. Okay, so how do we prevent this? Okay, of course, if we prevent SARS-CoV-2, this is somehow same with the prevention because this is also droplet. So stay at home. Okay, so eat right. Okay, avoid touching your surface. Okay, keep surface clean and wash your hands. So get vaccinated. So this is usually yearly. Sandali lang, I have to answer this phone. Hello? Apa. Ay, apa, Doc. Dr. Rafael. Apa. Apa, Doc. Yes po, yes po. Si Gilmore po, opo. Ganciclovir. I think it, yes, doc, yes, doc. Okay po. Yes po, yes po. Oo, doc, mahirap, mahirap. Mahirap talaga, doc. Opo. Yes po. Yes po, doc. Ma yeah, Val. Yes, doc. Yes, doc. Yes, yes, doc. Oo, yes, doc. Mahal talaga po. Yes po. Oh, no. Yes, yes. Yes po, yes po. Yes po, doc. Opo. Yes, doc. I will make a way po. Yes po, doc. Okay, doc. Thank you very much. Yeah. Thank you po, doc. Kaya ini-startan ko na rin siya ng antiviral, antiretroviral. Yes po, doc. Thank you very much, doc. Opo. Thank you very much, doc. Yes, doc. I will coordinate with Globo Asiatic, doc. Globo Asiatic usually also gives. Yes po, doc. Yes po, doc. Yes po, doc. Yes. Opo, doc. I will. Okay, doc. Thank you very much po, doc. Salamat po. Opo, opo. Ingat po kayo, doc. Okay, sorry. I have a patient with cytomegalovirus. Okay, has infected his retina. Okay, and he's nearly going blind. So they have to be fast in treating this cytomegalovirus retinitis. Okay, because it may lead to permanent blindness. Bata pa naman yung pasyente, only 24 years old. Okay, so that's why yung ophthalmologist called me. Okay, so yeah, smile during this pandemic. Kahit mahirap, we have to smile to para kahit somehow bumaba yung stress natin, because if we have a lot of stress, we are prone to any form of infection, whether this is virus or bacteria. Yeah. So of course, get vaccinated. So when the Sinovac or the COVID-19 vaccine is available, you have to receive whatever available vaccine is, you have to receive it. Huwag ka nang pili na iba because you do not know when this will be available. Okay, so there are three C's in preventing this flu. We have to clean, cover, and contain. Okay, so this is in 2018, the status of influenza here in the Philippines, around 45%. Okay. Kasi kasama ito sa surveillance of your influenza-like illness, okay, including your SARS-CoV-2. Okay, so antigenic chains, so we have what we call your drift, okay, and we have what we call your shift. If it's drift, it's usually minor, or you have point mutation. Shift usually drastic and abrupt. So this is the differences between your antigenic drift and antigenic shift. So minor change, major. There's usually point mutation. There's exchange of gene segments. So this occurs in A and B subtypes. This one shift all occur in A. Yeah. So may cause epidemics and they may cause pandemic worldwide. So these are examples of your influenza. So this is your influenza drift. You see this one, okay, changing the shape, okay, can hardly be recognized by your antibodies. So your new subtypes nagbabago lang, regular assortment, but the shape remains the same. Okay, so this is the after vaccination. So you have given antibodies. So these antibodies will now bind to your hemagglutinin so that there is latch. So maglalatch yung antibodies to hemagglutinin so that this hemagglutinin can no longer bind to the respiratory cells. So once there is antibody. So this once there is antigenic shifting, you see, it assumes several shapes. Ito yung unang shape niya. And once it assumes several shapes, it can no longer be identified or recognized by your antibody. Okay, so this is your antigenic shifting from flu coming from the duck or bird. Okay, so there will be genetic reassortment. So this genetic reassortment may go into pig. So magkakaroon ulit ng genetic reassortment sa pig. Ginamit niya yung pig. Okay, and this pig, okay, may transfer the virus, the new virus strain, to human. Yeah. So the new strain may further evolve to spread from person to person if also pandemic could arise. So kindly read over this one. Okay, so in 1918, there was a Spanish flu, H1N1. Yung H1N1 ito, nagkaroon ng genetic reassortment with the H and N with this human. So the bird to human transmission. So na-combine H1 and N1. Okay, so there was genetic reassortment and there was all 8 genetic segments to have originated from the avian virus 1, 2, 3, 4, 5, 6, 7, 8. So in 1957, we have the Asian influenza. We have H2N2. So the H2N2, okay, of the avian combined with the H1N1 of the human. So there is genetic reassortment and there was 3 new genetic segment. 1, 2, 3. Okay, so pag nagkaroon ng genetic segment changes, may iba yung shape ng heme agglutinin. Okay, there will be assume several shape. 1968, there was a Hong Kong influenza, okay, H3N2 influenza virus. The H3, okay, of your avian binding with your human virus. So there's genetic reassortment, and there was 2 genetic change. One, the yellow one here, 2 genetic. Okay, and we do not know what will come next. Okay. So this is your genetic reassorting. Okay, so the period between epidemic wave of influenza A tends to be 2 to 3 years. Okay, so we have this happened in 1918, H1N1, 1957, H2N2, 1968, H3N2. So your Hini, we call it Hini, H1N1, sometimes emerged in 1977, although no epidemic materialized. 1977, H1N1 and H3N2 viruses and influenza virus have been in global circulation. Now let's move on to your avian influenza. So there are 15 HA subtypes found in birds, same with humans. Okay, only have been transferred to animals, H1, 2, and 3, H5 in human, H1 and 3 in swine, and H3 and 7 in horses. So the same pattern holds for new raminidase, 9. Okay, NA, known for birds. Ganun din naman sa human, 9 din. So only 2 of which are found in humans, so N1 and N2. So they do not appear to undergo antigenic change in the birds. Okay, bakit? Kasi yung birds namamatay rin naman. Brief lifespan. So walang antigenic change in the birds. Okay, so birds to humans. But birds, hindi. So the avian influenza ranges from inapparent infectious to highly lethal infection in chicken and turkeys. So yung avian influenza, pwedeng ma-transmit going to humans. Okay, yan. So your H1N1. So most influenza infection in ducks are avirulent. Pag sa ducks, usually avirulent. Okay? Pero they are highly infectious going to human. So that's why this infected poultry product, ay chicken, should be killed. Okay? O ikakal nila lahat ng, they have to cull, okay, this infected chicken. So, viruses of ducks multiply in cells lining the intestinal tract, ayan, and are shed high concentration in fecal water. So, when this fecal material dries up, it can be inhaled by a susceptible human, okay, where they remain viable for days or weeks, especially in low temperature. Ayan, punta yan sa bata. Okay, so this is also from ducks going into pigs. So there will be genetic reassortment in pigs. Okay, and pigs may transfer this virus to humans, and humans may also transfer this virus to the pig. Okay, so evidence support that models that pigs serve as a mixing vessel for reassortants. Okay, so this is the pandemic strain in 2009, was genetic reassortment. In 1997, in Hong Kong, first documented infection in humans by the avian influenza A. So that was H5N1 in 1997 in Hong Kong. So the source was domestic poultry, so chicken. So in 2006, the geographic presence of this H1N1 avian influenza in both wild and domestic birds have expanded to include many countries in Asia, Africa, Europe, and Middle East. So outbreaks were largest and most severe on record. Okay, so this is your avian influenza. So you have the H1N1 in avian influenza acquired the ability to spread efficiently to be sustained among humans, either by reassortment or adaptive mutation. So this will result in devastating influenza pandemic. So this led also to pandemic. Okay, zero archaeology. Okay, so your history of your influenza, okay, have been documented. Isolation of H2N2, Asian flu, appeared in 1957 and was replaced in 1968 by H3N2 subtype. So you see, genetic reassortment talaga. And H1N1 strain reappeared in 1977. Reappeared, so nag-re-emerge. So there was re-emerging. While your SARS-CoV-2 bago yan, emerging yan, emerging. Pag re-emerge, dati na, bumalik ulit. Okay, so how to protect? Okay, is through your influenza vaccine. So it's available now. It's Fluarix. Okay, so galing naman sa GSK. So Glaxo produce your Fluarix, while the Sanofi is Fluzone. Glaxo. So this one, how do they do it? Okay, so they get the virus here, the virus, okay, the flu strain, and they inject into the allantoic fluid of the egg. So this will now multiply, okay, the viral multiplication, and they have to add products here, chemical, in order to inactivate this virus. So when it's ready, now ready to be given to individuals to protect this, to protect all of us from flu, okay, getting into severe. Okay, so everyone aged more than 6 months without contraindication should receive flu vaccine. So clinicians should offer or vaccinate at the end of October. So dito naman, usually it's become available here in the Philippines, March, April. So children who need those should get first dose as early as possible to receive the second dose 4 weeks later at the end of October. Okay, so this is usually in the northern and southern hemisphere. So other vaccines are usually influenza in other countries, usually intranasal. So in summary, so influenza viruses are the major respiratory pathogens. So your A is highly variable, antigenically, and most epidemic and global pandemics. So your B sometimes undergo antigenic changes and can cause epidemic. So your C is antigenically stable. So your A strains are also found in aquatic birds, ducks, domestic poultry, pigs, and horses. So the viral genome is a single-stranded negative sense segmented RNA. So you have two important glycoprotein, the hemagglutinin and the neuraminidase. So they determine influenza antigenicity, and human immunity. So minor antigenic changes in hemagglutinin and neuraminidase are known as antigenic drift, while major antigenic change is what we call your antigenic shift, result in new influenza virus subtypes, okay, because of the genetic reassortment. So because many different viruses can cause respiratory infection, diagnosis of influenza infection cannot reliably be made clinically and relies on laboratory assay, the PCR. So immunity to influenza is long-lived and subtype specific. So only antibodies to HA and NA are protective. Okay, so this is the summary. So both inactivated and your live virus vaccine exists but are continually being rendered obsolete as new antigenic variants of influenza virus arise. So kaya nga yearly, because new antigenic usually they usually know this antigenic type serotypes at the October, November, December here, and once they knew this type already infecting this 2021 flu, okay, then they already made this, okay, and this will be now available here in the Philippines. So antiviral drugs exist, but resistant virus emerge frequently, especially M2 ion channel inhibitors. So a highly pathogenic avian influenza virus like H5N1, what happened in Hong Kong in 1997, causes a sporadic human infection but has not acquired the ability to human transmission. So naagapan din naman. Okay, so that is your influenza virus or your orthomyxo viruses. Any question? Do you have some questions? Okay. Any question? Okay, your